Hello, everybody, and good evening again. My name is John McGoldrick, and my pleasant task tonight is to introduce Dr. Elliott Sigal. He's going to speak to us, as I'm sure you know, on drug development and cancer. One morning, some number of years ago, I was sitting at my desk, and a memo hit my desk. It was a memo about a new employee who looked full of promise to me. Years later, Elliott Sigal stands before us today as Chief Scientific Officer of Bristol Myers Squibb, as well as Executive Vice President and a Director of the company. But he is much more than that. Elliott is truly one of the great stars of this era in the development of new cancer drugs. He has already accomplished the really remarkable feat of bringing 14 important new medicines to patients during a relatively short tenure. It's really quite amazing, and people in the industry, those who are here and those not, will tell you that that is so. 4 of those are major cancer drugs, and he is widely recognized by his colleagues and by his competitors globally for his achievements in developing life-saving and life-bettering drugs. He has many awards, many papers, and many accomplishments, and I think you'd better hear from him than from me, or we'll be here all night if I started to go in on those. With all of these accomplishments, yet you might see Elliott on the street, or in the halls of BMS, or over in the dining room sometimes, and not realize all of it, because Elliott is quintessentially the team player. Quiet, thoughtful, not seeking the limelight. As a managerial principle, and he can tell you at length about this, he believes strongly in teams as a road to success. He is certainly a strong leader, adept at that decisiveness in the midst of ambiguity required by the job he has. But he always credits his teams for his successes. With Elliott, it is always we, not I. Elliott brings many special talents to the fight against cancer and other diseases as well. His engineering and mathematical background. He's a PhD in engineering with a dissertation in stochastic mathematics on the shortest root problem. We won't talk about that, at least I won't tonight. His MD followed, and a clinical practice thereafter at the bedside in the chaos of HIV/AIDS in San Francisco in the very beginning of the epidemic. He saw that up close and personal, and patients up close and personal. His work at the bench on hemochromatosis, resulting in the cloning of a gene to address iron overload disease, an important breakthrough, and his success at a biotech company, a small biotech company initially. It's a great variety of experience that he brings to bear. Different disciplines, different kinds of work experience. This variety of skills and experience no doubt lies at the heart of his success in applying the intersections of multiple disciplines: biology and medicine, informatics and genomics, biotech and pharma. These all have yielded these intersections, these life-saving and life-bettering remedies I speak about, and that have affected scores of patients who will never know his name. Tonight, as you know, Elliott will talk with us about drug development and cancer. Roughly 30% of Americans will suffer from cancer at some time in their lives. Still more will be profoundly touched by the disease in friends and relatives. Some of you in this room may know the sometimes exotic-sounding names of some of these medicines. Erbitux for colon cancer, Exempra for breast cancer, Sprycel for leukemia, Yervoy for melanoma. Tonight you meet the person who is more than anyone their author. We could not have a better and more accomplished leader through this subject than our fellow friend of the Institute, Dr. Elliott Sigal.
Thank you very much. Wow, John. Maybe I should just take questions. That's an extremely kind introduction, John, and I really appreciate it. I'm delighted and honored to be in front of other friends tonight. As you heard, I took a slightly unusual path to medicine. And for the mathematicians perhaps in the audience, that's why I had to study the stochastic shortest route problem. From the time I was 12, my father, who was a PhD organic chemist, had urged me to go into engineering. He saw a young son who was interested in math and science and big projects and big things. And this is a lesson to parents. He dissuaded unknowingly any interest in going to medical school. I've thought long and hard about this. He was a PhD, and I don't know if you still have these dynamics between PhDs and MDs. That's how I solved my life's tension. But it also is possible because his brother-in-laws were all practicing doctors. I was 17 when my father died of cancer. And one of my brothers and I were sitting at his bedside. And my dad said, why didn't either one of you consider, as you think of your career, going into medicine? And we were just floored. He didn't realize how parents can influence their children by their biases. I went on to college and stayed on my engineering track, and my brother went on to get a PhD in organic chemistry, and in fact he ended up doing research at Merck, and he is one of the pioneers of the research work in HIV/AIDS, the protease inhibitors that led to the control of that awful epidemic in part. By the time I was 24, we had lost our mother to breast cancer. With both my parents taken by this illness, I started to think again. Why not medical research? Why not the discipline of engineering and mathematics? Why not skills applied to medicine? That was my chance to help and to give back. And my training at the University of Chicago and California, San Francisco was all geared towards medical research. I began treating patients, then developed research programs, spent some time in academia, but I went on to industry for the big projects. For the last 20 years, I've had a career in pharmaceutical research, and I've had the privilege over the last 10 years to direct the development of new medicines in the variety of areas that John described. So what I'd like to do tonight is touch briefly from the patient perspective, hopefully, of how far the industry has come in the journey of our fight against cancer. We have a lot to do. Breakthroughs come in incremental steps. I will describe one patient experience that provides a glimpse into the breakthroughs yet to come. And then I'll bring back the discussion to the issue of academic collaboration. Improvements and innovations in both the detection and treatment have provided real gains against cancers over the last several decades. Age-adjusted 5-year cancer survival rates in the US have increased by more than a third since 1975. Certainly we've been able to control or cure very few cancers, but incremental progress in different therapies offers hope of improvement in lives. Some of this is due to detection and prevention, which is so important, but about half of the advances have been attributed to therapies that are being developed. Oncology remains a major unmet medical need, one that proves very challenging to this day, and is research that's not just done at the NIH or in academic environments elsewhere, or in small companies or large companies. People don't always see, unless you see what I see, that this is an important ecosystem of collaboration. There have been some major advances in drug therapy in the last 20 years that I'd like to highlight in 3 general groups, and then I want to give some examples of each of these. First of all, it was thought for 20 or 30 years that if you could just choke off the blood supply of a rapidly growing cancer, you could control that disease. And Avastin from Genentech was a monoclonal antibody directed towards a very interesting protein involved in developing vasculature and is used in a variety of cancers. These are anti-angiogenesis inhibitors. There's an array of what we call targeted medicines. As the Genome Project, as our molecular understanding of cancer has evolved, we have been able to say this particular gene or that particular gene is involved in an important pathway. Sometimes we're able to personalize the treatment by saying this particular cancer has an expression of a gene or a protein, develop an antibody or a drug towards that, and have a high probability that that patient may have a favorable response. Herceptin is used in breast cancer against the protein HER2. That's a Genentech product. Novartis changed the landscape of chronic myelogenous leukemia with Gleevec, directing a drug to the very basic genetic abnormality, a gene called ABL, that is overexpressed and needs to be inhibited. Erbitux from our company directs against an epithelial growth factor receptor and is approved for colon cancer. We know it doesn't work in certain types of cancers that have an altered gene called KRAS, and so we can test the patient beforehand to determine whether that will be efficacious. And there are a variety of agents of that type. Now with the Genome Project in cancer advancing, the government has built a genome-wide atlas of abnormalities in different cancers. And we can detect lung cancer abnormalities and regroup them by the gene abnormalities. And a recent drug, Xalkori, directed towards a kinase that's abnormal in lung cancer, may affect only 5% of patients, but we can pick out those patients by a genetic test and administer a targeted medicine. Quite recently, and something I'll spend a little time with, we've learned how to harness one's own immune system to fight the invading malignancy. This has been a dream for over 100 years, to have the natural defense systems of the body elicit against the invading cancer. And there's been some breakthroughs, both in prostate cancer and in melanoma. The third advance that's worth noting is the use of natural antibodies that can home in on a specific cancer and deliver a payload of a traditional chemotherapeutic agent. And I'll say something about that trend because we're going to see a lot more of it. But that's an ability to target and to deliver a chemotherapeutic agent And we know too often that the problems with chemotherapeutic agents is they hit normal cells. So if you can target them better, you can have increased efficacy. And over this time period, we've seen a lot of traditional chemotherapy continued to be developed. Roughly 30% of the biopharmaceutical industry's portfolio, and that represents a total portfolio of $50 billion a year, is focused on research in oncology, with tens of billions of dollars in funding coming from the private sector biotech. I'm confident that we should see greater breakthroughs in the future. And before I leave this slide, I'd like to recognize a laboratory here at the Institute, Dr. Arnie Levine's lab. In the study of the molecular biology of cancer, he was one of the first to elucidate a pathway of an oncogene, P53, which is seen in a whole host of tumors, and continues to contribute to the literature in molecular biology of cancer. He couldn't be here tonight because he's being honored at a major cancer society this evening. So there are three examples I'd like to use to bring some of these developments to light with an idea of what we might expect in the future. There's an explosion of gene and protein information about tumors, and we are therefore moving from looking at tumors under the light microscope and analyzing the gene signature or gene makeup, and we're reclassifying tumors. This has increased our understanding of which targets to go after in pharmaceutical applied drug discovery. It also gives us information to personalize treatments because we can keep track of a molecular signature and try to match patients with therapy. As you just heard from the introduction on the other slide, we are using precision targeting of cancer cells with antibody directed cytotoxics, and I'll give an example. And then one thing that is generating a huge amount of interest, and at the upcoming clinical research societies, I think we're going to have a lot of breakthrough information about clinical trials of eliciting the immune system across tumor types to fight tumors. So let me say something about genomic information and moving from histologic to molecular classification of tumors. On the left is a diagram that shows what we all know is that tumors that arise from a site of the anatomy adopt the name of the cancer. Hence, we have lung cancer, colon cancer, or prostate cancer. However, on the right, just as an illustration, if you take the molecular signature, the molecular makeup of different lung cancers, you have a host of different types of lung cancers. And in fact, if you do the same with colon cancer or prostate cancer, you can find that, for example, in red, you'll have a group of patients with a certain genetic signature that seem to have a lung cancer that behaves similarly among that group, but different than other lung cancers, either to treatment or natural history. And sometimes that signature tells you that that lung cancer is more related to a colon cancer, shown in red, or even prostate cancer, than other types of lung cancers. So this means that we are beginning to understand how to stratify patients or personalize treatment, as well as target medicines better. Let's use an example of use of gene analysis or genomics to stratify or subset cancer patients. In the middle is a picture of a genetic test, and typically the reds and the blue colors mean genes are turned on or turned off. And you can start to see a pattern that allows you to distinguish one patient's tumor from another. And on the far left is a group of patients without knowing a priori whether the patients will respond to therapy. You might know that in a certain population, there are about 45% of patients in that population that may benefit from therapy, and they're illustrated in orange. The question is, can we know before we subject them to a given therapy? Or can we select a different therapy or save them from the therapy and do something more appropriate? So a genetic test can separate into a population where you'll have a probability of you'll have a selection of maybe 60% of the patients that you might treat, and you won't treat with that therapy the other 40% of the patients. But if you've truly enriched the upper population on the right to those that will benefit from therapy, shown in blue, instead of having a 45% benefit over a population, you have a 75% benefit from therapy. And the other patients, if your test is good enough, show you that you should save that patient that type of therapy and consider alternate therapy. These numbers are real. They are actually extracted from our experience with a gene test for Erbitux for colorectal cancer. And a test is available to test the activity of a gene called KRAS. And if it's mutant, we know how to apply that therapy to that group of patient. And if it's not changed, we know a different therapy for that patient. Next, I'd like to say something about the class of drugs that have just started to appear but are in the pipeline called antibody directed cytotoxics. Now, this is a relatively old idea, but the technology is getting better, and sometimes it's worth staying with something 20 or 30 years until we get it right, and that actually is an example. An antibody is a natural protein that can be produced in an organism, a human or a cell system, and shown in yellow are the parts of the antibody that are the business end where they can actually attach very specifically to another protein. That's how the body defends itself. It recognizes abnormal proteins like invading organisms from bacteria or viruses, and antibodies can be generated. This can happen. You can now in the laboratory make an antibody to any specific protein, and therefore you could have a homing device. If you take a protein that's specific to ovarian cancer or to breast cancer, you can very specifically have that antibody go to that specific tissue. The linker in the middle is a chemical that attaches just the right way to the antibody and takes along with it a payload, which could be a conventional chemotherapeutic agent, that if given to somebody systemically has no specific targeting and can damage normal cells as well as cancer cells and has a huge set of side effects. The idea is to have the right kind of linker that holds on to that payload and releases it at the right time. Because you can see that the diagram of a cancer cell on the left shows specific proteins, which here are called surface antigens. If the antibody is directed in a very specific way to that particular malignancy, this whole set of weaponry can be targeted to a specific site, and if that linker holds on to the cytotoxic agent until it occurs, till this attachment occurs, you don't have as many side effects. And if it allows that cytotoxic agent to be released at the right time within the tumor cell, you have the effect. And then the third area I'll spend a little time on is harnessing the immune system to help attack cancer. This is a relatively new development along an old idea. Actually, I found a reference that's about 100 years old of a Dr. William Coley of New York City, who observed that patients that were infected, for example, a strep throat, and also had a malignancy, rarely, but occasionally, developed an immune response where when they got better, their tumor shrank. Now, this was old folklore for many years. And as science developed with a greater understanding of immunology, the hope was that we could very specifically elicit an immune response so the patient's immune system could fight cancer cells. We are now delivering this in clinic, and the first such drug is available called Yervoy for melanoma, which has classically been thought of as an immunologic tumor, that is, it could be modulated by immune factors that are administered. And we're seeing that in a small subset of patients, there's a potential for cure as defined with no evidence of disease after 5 years. Now, we don't know how long this is going to last. We have some patients that are out 7 years. But unlike many therapies that delay in a metastatic situation cancers ultimate fate. There's a subset, although small subset of patients, that have a very durable response, and this is generating a huge excitement in the field. And with these probes, that is new therapies, we're trying to different tumor types because you wouldn't expect that this would necessarily be limited to skin cancer. And we're finding that the old classification of what type of tumors are responsive to immune factors is being rewritten. And probably many tumors may well be susceptible to this approach. So let's delve a little bit into the mechanism. On the far left are two diagrammatic cells, one called an antigen-presenting cell and one the activated T cell. T cell, as you recognize from the immune system, a certain set of immune cells that can attack evading organisms, often after they recognize that a material is in the body and is foreign. That presentation of whether a protein is self or non-self is produced by cells in the body called antigen-presenting cells, because our immune system typically doesn't attack our normal cells. Our immune system is able to tell what is us and also able to tell what is foreign. So that interaction between an antigen-presenting cancer cell and an immune cell called T cell can lead to activation. An activation of a cell can make them, as diagrammatically shown here, angry. They emit chemicals, they emit proteins, and they can attack and destroy other cells. I zoom in here on an antigen-presenting cell and on a lymphocyte, a T cell from the immune system, to show that people have worked out the molecular mechanism by which the antigen-presenting cell actually interacts with proteins shown on the red of the T cell. And the abnormal protein is couched in the middle. And when that connection occurs, it's possible that that can activate the T cell. So the mechanism here is some foreign protein being recognized, the immune system has a chance to be turned on. It turns out that a second signal is required to make this happen. And this research led to identifying a connection between, on the right, a protein that in a non-informative way is called CD28, reacts with two proteins called CD80 and CD86. And when that connection occurs, if the other bridge has been formed, the T cell says this is a foreign material and I will start to fight as a member of the army. Now what happens, and we don't know exactly why, but cancer is very tricky and is able to trick the immune system to say, stop this reaction. And it actually produces, it causes the immune system to increase the production on its surface of another interesting protein called CTLA4, knocking out the interaction between the upper bar, CD28, and the CD80 and CD86. And what that does is put the immune system in check. And the marvelous discovery of scientists many years ago was to say, if you can block that CTLA4 from doing that, you release the brake that the cancer puts on the immune system, and you allow the immune system to do its thing to clear this foreign material that is a malignant cell. And that drug that we tested for many years is called ipilimumab. Unfortunately, we didn't have to teach many people that name because they give other names. It's now called Yervoy. It's been marketed. It was one of the first trials that showed a survival advantage to standard of care in metastatic melanoma. And I'm going to give some examples of using Yervoy on the patient level. But before I leave this, I'd like to say that there are other interactions in this mechanism between an antigen-presenting cell and the immune system. And there are other antibodies that have been developed to interrupt these interactions. And there's multiple connections here The excitement of researchers is there's multiple ways to manipulate this system, multiple conditions probably to use these different therapies, and a possibility to combine them to be more effective. So let's take this to the patient level, and I do tell people that this can be very graphic, but I want to make a real human point. This is the pictures of the back of a male patient who allowed these pictures to be taken when he had a malignancy that was bursting out of his body. He had a melanoma removed several years earlier. He was treated many years ago, failed multiple rounds of therapy, and he appeared in clinic with this picture on his back, of his back. A physician at Sloan Kettering was working with our scientists and had ipilimumab, the antibody that I mentioned, in an experimental setting. And he began to deliver this to this patient because there was no other option, and that's typically the way we start our experimental research in cancer. And after about 12 weeks, it looked worse. And this is significant because there were many researchers in the field that were beginning to test hypotheses here. And the classical wisdom in cancer is that if you give a medicine and it starts to shrink, you continue. If it gets worse, you stop. Well, if you think about what might be happening here, if you're eliciting an immune reaction, you're going to get some swelling, you're going to get some natural reaction that's going to look worse before it looks better. That seems simple in retrospect. There were many companies and there are a few companies and other researchers that missed the fact that this is probably the expected reaction. Dr. Wolchuk at Sloan Kettering continued with this patient, and in 2 weeks, there looked like some improvement. And in some cancers, I'm showing just a metastatic, you could be quite pleased with this after 2 weeks, and sometimes that's all you have, and sometimes you put that in check. I will say we're looking at this gentleman's back, but this tumor was throughout his major organs as well. At week 16, this began to fade in a convincing way. And remarkably, at week 72 and week 108, and today, many years later, there is no evidence of cancer in this patient. Now, as I tell employees, and I told John at one point, doctors in the old days would have called this a miracle. It is a miracle. Today, we're required to call it an anecdote. And we can't be sure until we do rigorous clinical trials that this is going to happen to more than one patient or to be more than a rare event. But this is remarkable, and it happens on a repeated basis. We can now say that through 2 randomized phase 3 trials that have been reviewed around the world. And what we produced in such clinical trials is what's called survival curves. They're Kaplan-Meier curves. I had the benefit of studying statistics with Dr. Meyer at the University of Chicago, who developed the statistics and the mathematics to describe from a clinical trial how you could represent a statistically significant difference when people are dying at different time periods by treatment A versus treatment B. The blue line is patients that are getting standard therapy, dacarbazine, DTIC, plus every 3-week shot of Yervoy or ipilimumab, versus the same treatment, dacarbazine plus placebo, that's shown on the lower line, a light gray. And this is a Kaplan-Meier curve that goes out months, almost, well, there we have 4 years of data. Typically, patients with metastatic melanoma, which all these patients were, that had failed any therapy, by and large die within a year. What's remarkable here is the separation of these 2 curves with hundreds of patients being tested that persist out to 4 years. Typically, a Kaplan-Meier curve, as I mentioned before, comes together after a period of time. You delay the process and then you can't affect the process after that. The fact that for a subset, now understandably we'd like that to be a larger number, but a subset of patients, about 20+% of patients, there is a durable, long-lasting, and hopefully longer than 4 years, we know it's longer than 7 years for these patients, effect. Many of them have no evidence of disease. This is remarkable, gives us a great deal of hope. This line, to make it more dramatic on the patient level, doubles somebody's chance of being alive at years 2, 3, 4, 5, 6, and 7, doubles. And our challenge as researchers now is to either identify that subset of patients, the 20%, so they get the medicine and know there's going to be a high chance of responding, or more importantly, increase that percentage from 20% to 30 to 40 to 50 and beyond. And we have evidence in research that if you combine therapies, even radiation, standard chemotherapy, or other agents like I showed before that modulate the immune system, we have a chance of increasing the percentage of patients that will respond. And our research is also actively looking at just not melanoma, but other tumor types, because this is not necessarily, like anti-angiogenesis that attacks the blood system, limited to a certain tumor type. One more just as compelling story, and I'm allowed to use her name, Sharon Belvin, who I met recently, was in some of our early clinical trials. She too had a lot of sunlight exposure and had something on her skin that was alarming. Shown here is her husband and her 2 children. But in actuality, years before the children were born, at the time she and her husband were getting married and her dress was being fit, the dressmaker noticed something on her skin. She went to the doctor. She had to have it treated. It was melanoma. She was a brave woman and wanted to go ahead and get married anyway. Her husband to-be agreed. She was sick throughout the honeymoon because of chemotherapy. She also came to Sloan Kettering in New York, failing all kinds of therapy and having systemic spread of melanoma in almost every major organ. She received ipilimumab, and now 7 years later, she has no evidence of tumor. It is remarkable that patients who had metastatic melanoma are able to go on to have any meaningful life, let alone have a family with 2 children. We are now collecting the stories of what we call ipilimumab babies, if you will. It's a wonderful human aspect of being in this field. So I'd like to come back up onto the level of what it takes to develop drugs these days is in the ecosystem of academia, government funding, small companies, large companies. And I'm a big believer, as you heard earlier, in collaborations. And some of our best breakthroughs come from the right type of collaborations. The whole story of ipilimumab comes from some academic scientist who modified something that some Bristol scientists had discovered, who then found a biotech company that would take the high-risk road of developing it to a certain stage, and then a larger company, Bristol again, merging with that smaller company and producing a large program. We are a globally interconnected world. The science is more and more complex. Regulatory environment is very, very demanding, appropriately so, and we're all resource constrained. So what is happening now in the field, especially since the financial crisis where smaller companies are struggling and academic labs are struggling, is to revisit the interactions that industry can have with academic groups. And there's a whole range of these that we're working on. We're actually funding a fellow, Dr. Elke Markert here in Arnie Levine's lab. We're funding Vanderbilt in Parkinson's disease. We're funding UCSF research in Alzheimer's area. And we have a whole network of academic collaborators in immuno-oncology. And we're able to learn, and other companies are doing the same, about new targets, new validated ways of therapy, new possible drugs that people are discovering in different parts of the ecosystem. And we're able to speed the translation of the science through clinical development. So I'd like to close with a lesson that I learned in the early days of medical training on the wards at the University of Chicago. I was going on rounds with the attending professor and what I remember is the J ward of a now closed hospital over 30 years ago in Billings Hospital. And one morning my professor told me to go into a patient's room to observe and then come out and report what I saw. So I talked to the patient. He was a young 38-year-old man. He was well-nourished, very muscular, had a good head of hair. And the only thing he complained about is when he was brushing his teeth, he was bleeding. His gums were bleeding, and he knew this was bad, but he didn't know what it was. So I had reasoned just from the history and physical that this was some kind of platelet abnormality. The giveaway for a third-year resident is I was on the hematology ward. But this is the way you're trained as a doctor. And I came out, and the professor asked what I would see and what I thought. And I said, I see a young man is full of life, but well-nourished, has bleeding in the gums, and I suspect some kind of platelet abnormality. And I said, which most medical students don't say, I said, what do you see, professor? And he says, well, I see a young man who is full of life, but who will, before too long, become emaciated. He'll lose his hair. His complexion will turn sallow. The energy you see in his eyes will vanish. Eventually, we'll put tubes in him again and again to see if we can stretch his life for just a few more days or weeks. I see a patient we can't treat. This man had already been diagnosed with blast phase CML, or chronic myelogenous leukemia, which in 1981 was a diagnosis of a painful death, almost universal. But what I learned was that what may be true one day can be transformed into something else the next. It may take time, even what may seem like an eternity. But the right minds working on seemingly intractable problems can change the world. Drugs such as Gleevec and Sprycel now keep patients with CML living normal lives for years, and we expect decades. We've come a long distance in the understanding more about the complexity of cancer. We have a lot of rich material in science to work with. We must find a way to deal with the regulatory hurdles or the financial hurdles or the collaboration hurdles, or the cost hurdles. There's hard work ahead. Each of us in cancer research are contributing, and I've been privileged to work with a team of people that have made some contributions and will do so for many years. And I wanted to share the story with you tonight. So thank you very much.
Elliott had said he was going to try to stop a little early. Not all of our speakers do that, as you know. So the questions can come from the audience. Elliott and audience, go ahead.
You don't have to have a question. A comment would be fine. Or yes, Don?
What would you say would be the most difficult? I'm sorry. Of all the tumors, keeping with the organs, if you will, for our understanding, what would you say would be the most intractable tumor right now to be working on? Is that a fair question?
Well, there's an unfortunate long list. I'd like to see more progress in brain cancer research. I think we're not done with breast cancer, but we've made major, major strides. We have trouble in ovarian cancer just because we can't pick it up as quickly as we would like, so there's a lot more detection, early detection work to do in that area. We've controlled some cancers in an amazing way, like testicular cancer. But I think if I had to pick something that, of the major 4 cancers, I would say lung cancer. If you can detect lung cancer early, you can cure it with surgery, but most of the time patients present and it's not surgically curable, and the treatment afterwards has improved, but it's not very good. So I'd like to see more advance in, off the top of my head, in lung cancer and in brain cancer. I will say there are a lot of smaller diseases we don't always hear a lot about, like mesothelioma, which is a horrible complication of asbestos exposure. That what we're trying to do is to find out some molecular clues anywhere we can, and then see if they apply to some of these smaller important opportunities. Yes.
So I've seen some companies are starting to.
Where are we? Oh,
Over
yes.
So I was just wondering, for what percentage of cancer patients is there actual genetic testing done on the disease, and at what stage?
Yes,
I mean,
we're in the infancy of this area.
if someone's already been doing chemo, do you really wait to the last possible moment to try an experimental therapy, or?
Oh, I'm sorry. I thought you may be asking 2 different
Yeah, I have
questions.
2 questions.
Do we routinely screen for genetics? It's being done more and more. In all fields, we're interested in looking for ways to predict who would respond. This is actually classical medicine. I mean, your physician tries to stratify patients by genetic history, family history, physical signs, etc. This has always been the way medicine can and should be practiced. So the problem with some of the genetic testing, we don't have enough validation for it. And I'm usually a voice for, let's make sure we understand what we're doing so we don't give somebody false hope. And you've actually read at some major institutions there's been some problems without people checking the quality of the analysis that's being done. But I know at most major cancer centers that you can think of in New York and Boston and MD Anderson and in Florida, most places are looking for genetic markers, even if it contributes to our knowledge, unfortunately not for that patient, but for populations in general. So we've struck up alliances with Moffitt Center in Florida that is genotyping and studying expression of proteins of all tumors that come in and keeping track of the databases that will allow us to tease out the relationship between therapy, genetic makeup, and response. And I think the movement in this country for electronic medical records will improve that. The enrollment in clinical trials for adults is extremely low. It's less than 5%. It would be helpful if patients did, if they felt they could, enroll in trials, because if they don't get benefit, it is often very meaningful to patients that they know somebody like them will. In the pediatric oncology field, it's a huge percentage of involvement. With regard to experimental medicine and who gets it, do you wait? You have to do the ethical thing. So we're not trying to experiment with somebody who may have other options that are proven. So we need to start typically at late stage and then gradually move earlier so that we gain confidence that we're offering something that's valuable to and ethically appropriate to a patient under this condition.
So sorry, just one
Yep.
follow-up to my first question. Pragmatically, can a person request that their tumor be sequenced, and
Well,
will
that's
insurance pay for
that's-
that, and so on and so forth?
I'm not sure insurance would pay for that. I do believe that probably at a good research institution it would be done. I'd have to get back to you with any more precise answer than that. Insurance would most likely pay for tests that have been approved by FDA, approved as being useful, but many cancer centers are sequencing so that we can improve the knowledge for patients in the future. Yes?
So this is related. Is the pharmaceutical industry or BMS, are you doing whole genome sequencing or exome sequencing of responders versus non-responders?
Well,
are
you
you
know,
moving towards that?
where people are moving towards that as the cost of DNA sequencing has gone down from millions of dollars per patient to hopefully a matter of thousands of dollars a patient. It's going to be possible. Most of our sequencing is done on a more targeted basis, but we're benefiting from the fact that the NIH and NCI have collected a whole series of tumors, and they're sequencing in a more comprehensive way and matching up clues of patients that we can then match to them. We started about, when I came to Bristol over 15 years ago, and it's taken longer than we had thought, trying to bank blood, for example, of all patients in clinical trials so that one day we'd have the record of those clinical trials and we can go back and do sequencing when it was more affordable. So it is happening more and more often. We do keep track of genetic markers, I would say, in 80% of our oncology projects now. Yes?
From a preclinical standpoint, can you talk about some of the larger challenges, maybe industry-wide, from getting, is it identifying the correct targets? Is it access to the linking technology you talked about? Where does the industry need to most develop, and what is most in demand there?
From the preclinical side, so all our testing in test tubes or experimental situations before human, the biggest challenge, I think, is whether the preclinical models act like humans. The real species that we need to test is human, but there's a lot of work that has to be done to convince yourself that that's a worthwhile thing to subject somebody to. And when you get excited about something in the laboratory, in fact, the common thing is, well, we now know how to cure mice, right? It doesn't translate into what we're all about. I would say that's good. Animal models are improving. Genetically altered animals that have more humanized systems can be tested. Humanized cancers, or cancers from humans, can be put into experimental situations. And that's all aiding it. What was the second part of the question?
The second part was the linking technology.
The link. So with regard to the antibody directed conjugates, so this is an old idea, and we have failed in the past and have gotten back into it because the linkers have been improved. So you have to get the right antibody, you have to get the right linker. I think the least important thing is the payload, but you have already things that are available that you know can be delivered. Getting the right component in all three parts of the system is important. The linker has been the big issue. Having the chemistry such that you attach to the antibody in the same place all the time. You can imagine it's typically a random connection. Gives the kind of variability you don't want in a therapy. So we're learning to use molecular technologies to engineer the antibody so that the chemistry can be done in a more precise way. So it's both, it's every element there.
Do you have any insight into why so few adults participate in clinical trials and what can be done to change that?
We tried using Lance Armstrong. That wasn't a great idea. Actually, it worked for a while. The Tour of Hope was ours. It seemed very exciting. It seemed to be the right thing to do. I've taken down my yellow jerseys recently. Education is extremely important. I think patient advocacy groups are very important. They have been very, very helpful with childhood childhood cancers, and there is a natural tendency to do everything you possibly can for a child. There are much more concentrated centers. There are much more specialized centers usually dealing with childhood cancer, not necessarily a community situation where that kind of research may not be available. That's just off the top of my head some insights. I think programs to educate people is the best I can think of, but I think it's a relatively low percentage still.
Are there any nascent or latent environmental triggers that we should be aware of given our way of life? The most obvious example that come to my mind is asbestos and tobacco and so forth, but these are very well established examples. But is the industry doing anything about those?
Well, the research community is doing more and more and needs to continue to look for toxins that or environmental factors that were not previously recognized. And the difficulty is you can do experiments to deal with acute exposure. The difficult thing is trying to figure out low-level chronic exposure. And unfortunately, this type of research generates hypotheses first in epidemiological studies. Think about how long it took the Surgeon General to say something strong about tobacco. Prevention is clearly one of the most important things we need to work on. Diet is extremely important, more than ever before. Obesity is a threat to every disease, including cancer. There's low levels of things in our environment that we don't really know a lot about and will be difficult to study, but that's ongoing. We as in an industry, and I'll just give you the challenge, it's despite what some people say, and my story is not about drug companies, my story is pretty typical about people that gravitate to research in pharmaceutical, about identifying what you can try to do to help a large number of patients. We don't often get involved, however, practically in a lot of prevention studies because it's so difficult to administer an agent and have it be safe to otherwise healthy people for long periods of time and do experimentation. So from a therapy standpoint, that's not where we can make the best contribution, but in terms of a community to support the kind of research for associations, it's extremely important and increasingly recognized. Oh, the director is going to ask me a question.
Well, thank you for a fascinating talk. I have two questions, actually. The first is there what's our understanding of what, in some sense, the immune system in just a healthy person, how effective that is in treating tumors?
Yeah.
And the second question is relating back to your title. If you could change anything in the landscape of pharmaceutical research worldwide, what would it be?
I know why you're the director now. You're asking the big questions. The very relevant question to the first point, what in the immune system? We're trying to figure out, scientists all over are trying to figure out, do people have a different set of primers in their immune system? Do they have a different gauge that's set? We just don't have ways to predict who's responding to these immunotherapies. We've only had experience with them actually working, although people have tried in the field for 20 years, and we were criticized for doing the first high-risk alliance, actually, with a small company to try to do that, but as a portfolio, you have to keep, taking risks as well as go down a well-trodden path. We're now studying patients to see if there's anything in their blood or their tumor that said something about the stage of their immune system when they had the therapy. We do feel that priming the immune system somehow with other agents, we're seeing some interesting responses there. So whether you can measure whether somebody will respond or not by the state of their immune system is just too early days. We do have some agents that target something specific, and that is a very crude measurement. If the target's not there, that's not the patient to give the medicine to. So the landscape, although this, it takes 15 years or so to develop a drug, really, It takes a huge amount of decision-making with a long period of time building on others' achievements. Given all that, I'm pretty excited, and I'm optimistic that we can translate this science into benefit. What I'm concerned about, and that's why I talked about the collaborations, is I think we ought to have more government funding in the basic research. We had to have more efforts to break down the barriers between the academic incentives and working with companies. And that's starting to happen. I'm on advisory committees at different universities, and things are happening in New York where it used to be you don't deal with the applied research of a drug company. We need the whole ecosystem working. If I had one thing to change, I would speed up that process. Maybe people are hungry, John.
But maybe you. Well, I think the time is up, and there would be more questions. I'm sure some of you may be able to capture Elliott after we stop this session. I just want to end it, Elliott, by saying on behalf of the Institute, thank you to you for
My pleasure.
coming,
Thank you.
and saying to the rest of you what I did not say in my introduction, because I knew you would see it in Elliott. He was a wonderful colleague when he and I were colleagues, and he is simply a wonderful man.
Well, thank you, John. Thank you very much. Thank you. Thank you very much.